VitaSite sponsors two different webinar series, one on human islet isolation and the other one on special topics in enzyme-mediated cell isolation. This is one of those special topics webinars, and I'm pleased to have Jay Hoying give a talk on human relevant vascularized tissue models. I think this is an up and coming in field, and I can tell you, I was in business development. I was at Boehringer Mannheim, and I saw the change that was occurring when genomics entered the field and the effect it had on forming a different type of biotech industry than what was done in the 80s. I think we're in the middle, and it was literally, as the overused term, an inflection point. Well, we're an inflection point right now with how to apply these new technologies to use cells for drug development, treatment, and whatnot. Clearly, we're in an exciting time, but also a risky time, because there's a lot of unknowns how well these things are going to work. So I think Jay's talk is extremely timely in that regard. Just FYI, I met Jay in 2007 when the Cardiovascular Innovation Institute opened at the University of Louisville. At that time, Jay was working with Stu Williams, who was the chief scientific officer, to develop in vitro microvascular models from adipose tissue. He then started collaborating with a Louisville-based company, Advanced Solutions, which focused on manufacturing issues, CAD drawings, and whatnot. And they had a strong interest in using 3D printers. And in 2018, he moved over to Advanced Solutions, where they created a new division called Advanced Solutions Life Sciences. He then moved up to Manchester, New Hampshire, because Dean Kamen recruited a whole bunch of new people to open up Army Biofab, where they got, I think it was a $75 million grant from the Department of Defense. And what I found was interesting is Army Biofab was actually like a skunk works. They were taking off-the-shelf technologies, and Rockwell Automation was doing the software, and the whole goal was to create tissue foundries. I haven't been up there in a while. I used to go to their meetings, but now they've got a separate building with, I don't know, 5 or 6 tissue foundries, where you put cells in on one end. It's a completely closed system, and the product comes out at the other end. It's interesting that Jay's in a position where they sell a bioassembly bot, which is a very sophisticated bioprinting instrument, and do applications. But he's also, as you can see from his abstract, looking at what's going to be the so-called new approach methodologies in human relevant tissue models. So without further ado, I will turn it over to Jay. And Jay, thank you again very much. Oh, 2 other things, ground rules before I get started. This is a open meeting. It's a Zoom meeting, so please mute your microphones. We'll have a Q&A at the end. And this session will be recorded and will be available on our website. And Jay has agreed to provide a copy of the slides on our website. So again, thank you very much, Jay, and I look forward to hearing your webinar.
Well, thank you, Bob, and not only for the years of discussion and conversation about tissue dissociation, etc., but for the invitation here. It's always fun to talk with you and to kind of share some ideas on how all this is moving forward. As Bob said, I'm chief scientist here at Advanced Solutions Life Sciences. I was in academia a long time before then. I'm a life scientist by training. really with emphasis on tissue biology and tissue vascularization. Here at Advanced Solutions, I oversee an applications lab up here in Manchester. And really what we're doing is using our technology base as well as other technologies to work with others to bring forward solutions and applications involving building tissues, primarily human tissues. And this is in the case of either tissue modeling for disease, modeling, biological discovery, drug target discovery. But as we'll talk about a little bit, if I build a tissue for a disease model, for example, for an in vitro assay, um, it's the same approach really in building a tissue for a therapeutic. Now, obviously, there are regulatory concerns that are different, but fundamentally at the biological level, in assembling a more appropriate representation of the human biology for a model, I'm going to be doing the same thing for a therapeutic. So while I'm focusing here on models, I think it has broader implications, and we can discuss what some of those ideas might be, how they translate back and forth. So there's been a lot of activity recently here in the past, I would say 5 years, I think, is when there's been, at least at the federal level, a lot more interest in action towards improving the situation that roughly over 90% of the drugs that are in the early development phase fail to make it to the clinic. A lot of reasons related to that, but a prominent reason is that it's just really safety and efficacy issues. And reflects in part that the most of that testing and evaluation is eventually done in animal models. And of course, we know that animal biology and physiology, while there are some aspects that are shared with humans, it's not 100% match. And so what is happening, I think, collectively over probably decades now, is that if a drug is shown to be safe or efficacious in a rodent, which is a very common model that's used in doing this drug development effort, that turns out that it's just not the case in humans. And so we'll go through a timeline here, but there was a concerted effort, interagency effort across also all relevant stakeholders in the industry and academia to really try to decide to do something about that. And the issue here is that not only are the animal-based models, poor predictors of drug success, as I was talking about, and certainly in some more complicated diseases, there's just not really a good animal model equivalent, even at a high level. But history has shown us that putting a drug through an animal model to evaluate safety wasn't necessarily required. So there are drugs out there that we're using that we know are safe that didn't go through an animal safety issue. Here in this example, aspirin was cited, and this is from an FDA roadmap to reducing animal testing and preclinical safety studies. And even the case of some animal models that are safe, it's the model predicted it should have worked in humans and it didn't, but in other cases here, fortunately minor or limited examples, the drugs were actually toxic in people while they were safe in animals. So there's a fundamental problem here that we don't have a very informed, predictive way to identify drugs that go in patients. Now, it has a lot of implications that we can talk about that I wasn't really going to address related to the cost of drug development because of this, reimbursement issues, healthcare, insurance, etc. So I'm going to focus really more on this idea of can we bring forth a solution that's less reliant on animal testing. Really, just to do a quick timeline here, all the way back to the late 1930s, when the Food, Drug, and Cosmetic Act came about. There are provisions in there that essentially required animal models as the safety efficacy testing. Now, in 1938, we certainly don't have the decades of history and experience we do now. I think, this was, I think, the best idea on paper, certainly to move forward. And it's dominated really drug development and safety and efficacy testing even to this day. Um, However, recently here in 2018, now, obviously discussions have been going on before that, but in 2018, there was an interagency, interinstitutional roadmap established for really starting to consider, okay, if we're going to move away from animal models, what does that next solution look like? So a roadmap was laid out here. What should the new technologies be, utilizations, what's necessary to validate and create confidence? In other words, if an alternative solution, a non-animal based solution, predicted efficacy and safety, how confident are we in that prediction? There was also an advocation for forming a formal interagency committee that would explore this and oversee this. That was formally established in 2000. This is the Interagency Coordinating Committee on the Validation of Alternative Methods. And it really spans a number of different federal regulatory and research agencies. It's housed within the National Institutes of Environmental Health Sciences at the NIH. And they continued with the roadmap development, ideas, etc., led to the modernization action in action in 2022, which is really within the the FDA and getting the FDA to recognize first of all this concept of moving away from animal studies, but also coordinately around this same time, there's induced pluripotent stem cell technologies coming out. There's regenerative medicine solutions that were emerging. There's regenerative manufacturing technologies and solutions that were emerging. Bob mentioned the Advanced Regenerative Manufacturing Institute which is, I think, the 9th or 10th Advanced Manufacturing Institute funded by the DoD that formed in 2017. All of these elements really represent fundamental changes in how healthcare is going to be delivered, how new solutions were developed, how they're paid for, how they're implemented, etc. So it was clear, I think, broad consensus that the FDA needs to really take a step back and look at how these new technologies, which may or may not fit within current paradigms of regulatory approval, need to be evaluated, and what can come from that. So in 2000, again, this new committee was formed about coming up with alternative methods away from animal testing. The FDA then gets starts to reevaluate how they look at new technologies and solutions in the Modernization and Action Act. And then in 2024, a more formal process with a series of guidelines and recommendations came out of the Interagency Committee for validating, qualifying, and what would a regulatory acceptance criteria look like. And collectively, these methods different from animal testing were called new approach methodologies. So keep in mind, this is just a year old, a little over a year old here. And really the process is starting here with this. Now, we've been working with others here at Advanced Solutions to bring forth advanced tissue models, as we call them, trying to generate better biology and what's going on. And there still remained a hurdle that this is all kind of fine and good, but until the FDA says, hey, we will accept these, there's just a large barrier or a large hurdle towards really supplanting animal testing with these new approaches. However, in April here of 2025, so a little over 2 months ago, the FDA announced a plan to phase out animal testing. The focus here was on monoclonal antibodies, but you can imagine there are going to be other drugs that will be relevant to that. The FDA calls out really that a lot of the acceptance here of moving away from animal testing is the establishment here of these human relevant models. So these are terms that are being championed. These new approach methodologies, or NAMs or NAMs. Really, it's starting to categorize, classify, qualify, and characterize what these new approaches might be to replace traditional animal models. We've been focusing, what I'm most familiar with is the human relevant models here and what do those look like. I think an obvious one is the in vitro human-based systems, and there's still some, say, single cell 2D cells in the dish, for example, that probably are still relevant here. But really the emphasis and the thinking is around, well, how do we capture better the disease biology, the tissue biology? There are new technologies out there, whether organoids or building microtissues or these microphysiological systems or organ on chips approaches. And so these collectively advanced tissue models here, I think, are one of the reasons why the agencies are saying, hey, I think we've got a possibility that we can substitute or supplant animal testing. There's also, particularly with the emergence, and I should say really rapid emergence of AI, whether, a variety of different artificial intelligent agents that can do a variety of different tasks, can take complex data sets and make predictions that are hard to do experimentally or for a person to do. Really, I think there's an acceptance here of a variety of different solutions. What we've been doing in advanced solutions is focusing on what we do best, which is these in vitro human tissue biology models. So with that context, really kind of get into some of the science and biology. The idea here obviously is we want to capture as much of the native tissue biology that's relevant to, in this case, would be drug testing, whether that's looking for safety or efficacy. I don't think anyone sets out, we certainly don't, to recreate the entire tissue bed or even organ. I think that's a vision that's some time away, but still one that is being worked towards. Again, probably just as much in tissue therapeutics as it is in tissue modeling. But the idea here is we need more informative models, collect enough of the biology such that the information we're getting from that model, depending on how we challenge it, is actually predictive or predictive enough of what might go on in the patient's tissue bed. A lot of components here with tissue biology. I think up to now with the 2D approach, the focus has been on what's our target parenchymal cell, whether it's cardiomyocyte in the heart, cancer cell in the cancer tissue environment, neurons, say, in CNS tissues. But of course, the tissue is not really just a cell with some accessory components around it. It's a system of not only the parenchyma, but as well as the stroma, the vasculature, in the spectrum of immune cell niches that are within the tissue. And then, of course, that tissue is communicating with adjacent tissue beds, as well as other organs and tissues in the body through a variety of different signaling pathways. So I think in order to really start to replace the physiology, so to speak, that the animal models were intended to really represent, we have to start building better tissues, and then we have to come up with ways to link those tissues together in a way that lets us evaluate those communication paths and look at the health and disease state as a systems problem, not a single cell problem. One of the things too that we've been focusing on, which I think is just as important, is how do you implement these models? So I may have a very relevant, a very predictive and informative tissue model, but if I can only do one of those experiments on one model in 2 to 4 weeks, for example, it's just not very useful to me. I can't do enough science to get enough information to really have confidence in what I'm doing. So we also spend a lot of time on implementing. I'm not going to talk about that today, uh, but we always have that in the back of our mind is as we work with others to build these tissue models, is it implementable? How do we bring it forth? How do we get it to where it can be used at least to some level of throughput? And then, how best to capture the better human biology that I'm talking about. Our focus has been on vascularization. It's reflecting somewhat my academic background, but also our expertise here in the represented by our broader team. And so I was going to focus on that perhaps and talk about what that really means to represent better the tissue biology, but by bringing in a vascular component. So we recently published a review on this process, and there are a lot of different ways, I think, to vascularize a tissue model. Vasculatures probably means something different to a lot of different people. I'm a microvascular physiologist by education, so I tend to come at this from a microcirculation physiology perspective. But there are really three general ways to do this. There's a fourth here that is really by creating perfusion channels through that, but that doesn't really capture any of the vascular biology that's relevant. So I'm just going to focus here on these other approaches. The tried and true, which has been going on for a long, long time, is it's just using primary endothelial cells, whether you're isolating those from tissue beds or deriving those from stem cells. Putting in an endothelial cell population, which is that cell that lines larger vessels and comprises the first layer of small vessels like arterioles and capillaries, to help a spontaneous generation of vascular components. This approach has some success, but it really hasn't solved the broader problem, which is the vasculature is a complex tissue bed, and in and of itself is more than just a single cell. Now, there are other solutions out there that are bringing endothelial cells in with other mural cell types, which I think is far more effective. Stu Williams, for example, in past webinars has talked about the stromal vascular fraction from adipose. That cell isolate, which is a heterogeneous mixed population of different cells, I think has pretty strong vascularization potential because of these other cell types that are present. The other approach, primarily in deriving organoids from stem cells. In other words, I'm going to build, say, a liver organoid or a brain tissue organoid, starting with a single stem cell and then inducing differentiation to these other cell types, often includes generation of vascular cells in that. What's nice is this kind of recapitulates some of the developmental biology that goes on. And so you get more complex tissues that are organized and structured in a more native way. And then our approach here is to use not single cells, but pieces or fragments of microvessels that we isolate from adipose. And this is really how Bob and our relationship evolved is primarily me asking Bob, how do I get these vessels out of tissue in a way that preserves my vessels and keeps the function intact. So just to kind of get to that and to show here, at least in part, that capturing relevant biology is more than just the cell types. It's also the appropriate structure, the spatial organization, and to create an environment that lets multiple different cell types do their thing, do the functional biology that what they do in the body. We're looking at an SEM here on the left of adipose. The lipids here have been stripped out due to the processing for the scanning electron microscopy. but I'm focusing there on one of the microvessels in there, and it's a complicated structure. Again, we say it's a microtissue in and of itself. It's just not a tube of endothelial cells, for example, and it's embedded there in the stromal space. Importantly, though, the vessels do more than just perfuse, which is certainly a very critical aspect of any tissue health and function. But the vessels serve as depots for other homeostatic cells within the tissue that move on and off the vessel space. And for us, that's the biology we want to capture. We think if we can do that in a way that's straightforward, easy to implement, then we can improve what the biology is doing in our models, and then therefore improve the predictability. So real quick here, we take adipose from discarded lipoaspirates, really starting with that same SVF kind of isolation approach. Only instead of digesting to completion, we do a partial digestion and then do a harvest, a separation, and a screening. The challenge here is, and again, reflecting the conversations I've had with Bob and Vitacyte's enzymes, is how do I digest this such that I get rid of the cells I don't want and I keep my vessel fragments in place and intact? In other words, here in the center lower panel, you can see our vessel structure is preserved. In the center upper panel is what the isolate looks like. There are a variety of pieces of capillaries here that are collecting. It's heterogeneous. We like that. Captures all elements of the distal vasculature. But importantly to us, we preserve the space. And if we go in and look to see what cells comprise each of the vessel elements, we see a spectrum of cell types, including those that reside in that perivascular niche. So we now have a single reagent that we think can capture at least some of that relevant vessel tissue dynamics that at the cell level. For example, a tissue-resident macrophage shown in green here in the confocal images that are part of the vessel wall. You know, working from this solution really as a foundation, we've been exploring what the impact is. What does if you include these vessel elements, what does better biology look like? And are we starting to capture what we want to in creating these human relevant models? Real quick here, just as a summary, these are fragments of microvessels. They have that native vessel wall structure. We've shown through a number of publications that it recapitulates vascularization. I won't go into that here. We think you can use them as if you're using endothelial cells. It's not quite straightforward. Part of what we do in-house is develop the protocols to implement the fragments. We have a number of partners that are using this in their models, as well as vascularizing tissue therapeutics. So we think we were onto something good here, and we've been spending a lot of time trying to vet that out. So I'll give you a couple examples here real quick to show you how that looks and really what the impact is on including a more complex vascular solution in your tissue model. The first one here is an adipose organoid. The goal here is to generate a pre-diabetes, pre-obesity model. Perhaps with the latest with GLP-1 agonists, this becomes less relevant, but nonetheless, I think it's allowing us to explore what kind of a non-infectious inflammation looks like, particularly in adipose in these kind of pre-disease conditions. We take MSCs, commit them to a pre-adipocyte level, and then we bring in our microvessel elements, which we call HAMVs, and then co-culture them together. Bodipi here is a stain for fat, so we can see our fat cells are maturing and look plump and healthy. Down below is the vascular bed that forms from those microvessels, which is very exciting and satisfying. But for us, I think when we include the microvessels, we now see a more stereotypical in vivo-like response to inflammatory challenges. In this case, we use TNF alpha. Whether that's looking at IL-1 expression or downregulation of some protective genes, PPAR gamma here, our vessels really when they're present, make a huge difference than just the adipocytes alone. And interestingly, with the vessels, there's now an upregulation of the insulin receptor, which adipose obviously is very key in insulin glucose metabolism. And so, with a simple addition of our vascular elements here, we're now starting to see some more relevant human biology. Again, we think that this kind of approach highlights or is an example of perhaps a model that might be useful to take the place of an animal, which certainly animals, rodents in particular, don't develop obesity at all the same way that people do. Maybe this will be useful. Similarly, in an in vitro thick human vascularized liver tissue, this is the case using primary hepatocytes, the stromal cells that make up the liver, and then our vessels. When we include our vessels, if you look at the top right here, the hepatocytes upregulate their drug transporter expression. We're still not quite where we are, how it is in the patient, but we're certainly getting towards that. We also, in this thick tissue environment, which we initially created to house a hepatocyte or carcinoma, to look at liver cancer tissue microenvironments, including the vessels, now give us a thick plug of liver tissue, human liver tissue, a more in vivo-like dynamic with respect to cytochrome P450 responses to toxins, in this case rifampicin. If you put this within a transwell system, so now we start to add another layer of complexity, which is interstitial perfusion and fluid movement through the system, then all of a sudden our tissues behave better, not unexpectedly. Urea production goes up, metabolic status starts to go up. But interestingly, when our vessels are present, the ratio of lactate to pyruvate production changes in a significant way. We don't quite know what that means, but to me, I like to show this because as we start to layer on different levels of complexity, we're starting to see more emergent, more in vivo-like behaviors in these tissue models. And an interesting dynamic we're seeing in some of our cancer modeling, again, a pretty straightforward model, where we put in a zone within the bed of collagen in a 96-well plate, for example, a zone of our vessels, and adjacent to that, a spheroid of glioblastoma cells, and let that system culture and evolve. And what we see is, and it's shown here in the middle panel, that as the vessels in red and the glioblastoma cells in green start to move towards each other and share space, we don't really see much of an interaction until we add a drug that targets the vasculature, in this case bevacizumab, which is an approved drug for glioblastoma as well as some colon cancers. All of a sudden now we see a change in phenotype of the tumor cell. In this case, the green cells now start to, and you can see that in the far right panel, they now start to attach and move along the vessel segments that were formed from our microvessels. So we don't see this when we add endothelial cells, only when we have our vessel elements in here. And now we're starting to see an emergent behavior that one is difficult to evaluate in a mouse model, say a glioblastoma, but two, it's really capturing some of what we think are those responses that are associated with a escape of a therapeutic challenge, which we know that clinically glioblastoma does typically escape and avoid bevacizumab treatment. Again, in a little simple form here, we can capture more of that human relevant biology, we think at least, and then it gives us an opportunity to start to explore some of these other more complex system-like behaviors. Also, real quick here, this is fairly new that we were seeing, which is that it's very hard for us to grow our vessels in a tumor organoid, unlike with the adipose organoid, unless we arrest cancer cell proliferation, and then we can very effectively vascularize, that's shown on the right here, those tumor organoids. So there's definitely some interesting vessel tumor cell dynamics that we're able to experimentally manipulate and understand better those relationships, despite the fact that we know that tumors and vasculatures have a very intimate relationship. We still don't quite know a lot of those mechanisms. So we're hopeful that we can uncover some new insight taking this approach. And finally, to really expand here on adding additional complexity to the biology in these models, we've been working to develop, using our vessel system, to build an in vitro microcirculation. So this combines microfluidic chips. So in an example of a microphysiological system, which is something that the FDA is interested in seeing come forth as a substitute for animal testing. And the idea here is that we know that we can grow new vasculatures from our vessels in a matrix. If we implant them, we know they connect and osculate with the host circulation, carry blood, and evolve into a microcirculation. So the idea here is let's replace the host circulation with a microfluidic system that has inflow-outflow channels, and we can grow our vasculature between those two channels, have those vessels connect to the channels, and then let the hemodynamics of intravascular blood flow drive the evolution of the microcirculation. And indeed, some of our early work, we're seeing that. This here on the left is an H&E of one of those, what we call the in vitro microcirculations, showing that we're deriving, this is after 2 weeks of culture with perfusion, we can derive venules, arterioles, capillaries. They carry beads, the vessels do. Now, this is in a thick tissue, millimeter thick tissues. So we're starting to represent what we think is a more, of a tissue volume that's relevant perhaps to some discovery efforts. We're establishing vessel walls that are mature. We're seeing a more heterogeneous type distribution of diameters that's representative of more of a in vivo-like microcirculation. And we see evidence here of maturation post-angiogenesis, again, that reflects deriving this. And so we're working here to refine this system to make it more throughput than what it is now. But we are starting to explore, well, okay, can we take one of our tumor organoids now and understand tumor vessel dynamics in the context of a perfused vasculature? Then that's what we've started doing here. And interestingly, as we can see, vessels here shown in green start to approximate and approach the tumor spheroid. What we have found is that the presence of the tumor actually accelerates the connection of the vasculature to the perfusion channels of the device. And so there's, again, this cooperativity that we're starting to see that that's exciting us, thinking we might have some new potential insights into how these tissue elements are communicating with each other. So with that, I'm going to change gears real quick and speak a little bit to what Bob was talking about when we first started, and that's what's happening around regenerative manufacturing. As I mentioned, or as Bob mentioned, in 2017, the Advanced Regenerative Manufacturing Institute was established in Manchester, New Hampshire, headed up by Dean Kamen, and a very effective, very robust team of his, working off of $80 million investment. It was recently renewed last year with an additional 50 million. And as progress continues, if it keeps at the pace it is, which has been fairly rapid, there'll be another 50 million added onto that. And the goal here is to stand up an industry for manufacturing therapeutic tissues. as I mentioned, building a tissue for a therapy or building a tissue for a tissue model has some of the core technology and approaches, which is why we located our applications lab up here in Manchester to really be adjacent to a lot of this activity. A lot of press has come out of this, a lot of success. In 2022, there was an additional 40-some million established to begin building a manufacturing infrastructure around building tissues based on what the Army and the Biofab Consortium has generated. And then recently in 2024, the whole area was designated a tech hub by the Biden administration and received another 44 million to really stand up the industry infrastructure now called Regen Valley, really building around this whole idea of can we put in place the industry foundation for generating regenerative tissues, and has a lot of implications in healthcare delivery, regulatory processes, policy making, etc. But the Regen Valley Tech Hub is really dedicated to addressing those challenges. And they're doing it really through a handful of ways. One is expanding the commercialization of the solutions. A lot of those are coming out of the Biofab USA consortium that Army oversees. What are the market dynamics related to that? And are hospitals prepared to use a tissue therapeutic in their healthcare delivery? What does the education workforce development look like? What do the skill sets need to be? Where are the people to do this work, the new industry coming from? And of course, some of the infrastructure around people working here. Childcare continues to be a problem, certainly here in New Hampshire, but obviously elsewhere as well. And allowing a nice kind of home work balance dynamic, etc. And then finally here, how do we determine whether this is successful or not? What do those metrics look like? What are those? And are we doing what we set out to do? So with that, I think I'll go ahead and finish. I think I've talked long enough. I thank you for the time, and Bob, thank you again for the invitation.
Yeah, thank you very much, Jay. And yeah, we can open it up for questions. All I ask is that, or I can start it off if there's no questions. Is there anyone want to ask a question and unmute themselves and for Jay?
Sure. I'll ask a question. This is Doug Arm. Hey, Jay, how are you?
Hey, Doug, good to hear you again.
You too. Thanks for the webinar. Some very informative stuff. And, it's a great, objective to try to get away from the animal testing. But I'm curious, you've shown some good results here on the, recreating some of the microtissues and organoids. And I'm just wondering about bridging the gap with the agency. What might convince them, at least especially on the safety side, perhaps, that this is the way to go. Is there a bridging period that's going to be needed where they're going to require doing both before it goes to clinical trials? What do you think on that front?
Yeah, I think it's a great question. And as part of what the FDA announcing earlier this year, that they'll now are looking to phase out animal studies and in place accept these human relevant models. In that document, in an announcement, is really a set of guidelines and criteria such that, okay, for us to accept a human relevant model that will demonstrate, sufficient safety and efficacy, it needs to have A, B, C, D, and there's a series of criteria. What's interesting, which I think is speaking to your question, is not a requirement to show that the animal model matches, or more perhaps a better way of saying is that your human relevant model matches the animal model. And a couple of reasons for that. One is they're trying to get away from animal models, so it's you can't really ask the group to do animal testing to validate a new model. And two, if that animal model is not predictive anyway, you're not really getting anything. So there's been a considerable amount of conversation about what does an effective predictive model, at least conceptually, have to look like and have to show before it's acceptable. And I just refer you to that document. There's a lot of interesting detail in there. It's not a trivial amount of work. It's going to take some years, I think, before that happens. You're probably going to see some successes in more simpler applications than in others.
No, it
Okay.
makes sense. Do you have a sense, as you say, simpler models, perhaps, which indications or which areas might be furthest along in that front? I mean, you've shown some good stuff there with some of the organoids, but what do you think?
I think most of these, right now, the FDA is paying a lot of attention to these biologics, these monoclonal antibodies, either the antibody fragments, et cetera. They're looking at a lot of immunology dynamics here. Organoids are I think, a key focus, primarily because I think that's where the technology development is most mature. Having said that, it's still not mature. We're still trying to understand what an organoid tells us based on what's there. Our approach is really developing methods to help people build, say, more informative organoid models, with the idea that as they're doing their validation and development work, we can partner with them to help them realize that.
Excellent. No, thanks. I'll turn it back to the floor.
Yeah, any other questions? So I can ask a question. As you know, Richard McFarland, who's at the Army Biofab Institute, when they did the 21st Century Cures Act, they formed the standards group for regenerative medicine.
Yeah.
And I think, and basically the FDA even went so far as, excuse me, to start looking to simplify how people submit regenerative medicine drugs into or therapies, I should say, to the FDA, where they wouldn't have to put as much paperwork. They could say they could cite a standard, and if they did it right, then things would go, and also the FDA knew what this meant. That things would go smoother. So my question to you is, what you've just presented is ripe, right?
Yeah.
For finding an application and getting many people involved in writing a standard, and even including the FDA, and that's going to accelerate the adoption of your model.
Yeah, I think that's absolutely right. With perhaps a little bit of color around, I think, or emphasizing perhaps this idea of interagency. So,
Right.
we've been working with Richard. Richard left the FDA after what, 20 some years, and helped drafting, as you said, a lot of the guidance around regenerative medicine products, and is the chief regulatory officer for Army. We've been working with him to bring forth the regenerative medicine solution, and how we approach the FDA. So really, I think two-part answer to your question. One is that the FDA has been far more collaborative, perhaps, than they've been in years past. They're recognizing that there's a lot of new here, there's a lot of innovation, and perhaps pre-existing approaches and guidelines and regulations don't apply 100%, and so they are very much are encouraging, and we're experiencing that firsthand. A lot of communication with the FDA before you even put in an application, right? So there's a lot of discussion, there's a lot of collaboration, such that we can bring to the FDA, hey, there's no such standard right now, but we think this is the
Right.
way we want to do the evaluation. Give us some feedback. And then, so that then starts to lay the groundwork for standardizing some things that just aren't in the place anymore. The flip side of that, just to highlight a challenge, is, and we're seeing this with organoid on chip situations, where you put an organoid in a microfluidic chip. There's effort, and I'm part of that standard evaluation committee right now, is how do you standardize using these chips in a way? Because the FDA says we need standards around that.
Right.
There are probably a dozen different ways to do a microfluidic chip, an organ on a chip solution right there. None of those are equivalent. All of those are telling you a different readout and a different assay. So we're actually in the perspective right now, okay, you may be using chip A, and that's going to give me a different validation, and it works differently than chip B. But for me to be able to evaluate the data, I need to see a standardized control. How did it report out on a drug that everyone's looked at? In other words, standardizing processes as opposed to components, just to try to get a feel for, how does the FDA look at data from a microphysiological system and say, yeah, I have confidence in it?
Yeah, I think that it's absolutely critical. And the best example that I could cite is how you really should be thinking about technology roadmaps or something of that
That's right.
nature, where you can bring people together. And the best example was actually the technology roadmap that they created in the 80s, where you have a non-competitive, everyone's working towards the same goal.
Yeah.
Because in essence, what you just said is absolutely appropriate. I mean, these models should be, and I've seen this, and I'm by no means a hepatocyte toxicology expert, but I've seen the whole field within a matter of years start adopting new models of transporters and all this stuff that's gone on. And we started just selling platable hepatocytes.
Yeah.
We made enzymes for hepatocytes were platable. And so, yeah, I think this is, again, an exciting time. And I think it's going to require people to put down their competitive hats.
Well, yeah, I think you're absolutely right. And we're seeing some examples of that. There's a consortial program involving a number of pharma companies, as well as institutions
Okay.
and kind of non-for-profit institutions, around coming up with better hepatotoxic assays and
Oh,
readouts,
really?
right? And it's called the OASIS program. It's run out of the HESI, which is an institute. They just finished, I think it was 5 to 6 year study looking at, with a panel of defined agents that induce different hepatotoxic responses in liver cells in 2D assays. They now are looking to do that same program and same evaluation in advanced liver tissue models, liver organoids, something like our liver microtissue, something like an organoid on a chip. It's a cross, company and cross agency consortium that's looking at this. So I think everyone wants to see a better solution,
Right.
from not only kind of an ideological perspective, but from a business perspective. If I'm exhausting my drug pipeline because my 2D models are no longer predictive, I need to come up with another solution. So whether there's altruism there or not, I think, necessity is driving a lot of this collaboration.
No, actually, that makes perfect sense why pharma is going to do this, because everyone benefits, and it really gets back to, and I'm sure you're aware of it, this old paper that was published in 2001 by the FDA called Innovation and Stagnation,
Yeah.
where they basically said that the reason that we're in a big fix a big problem with drug development is those models used to be, well, put it this way, they weren't as good models, but when they were developed within the industry, and the industry now doesn't do that.
Yeah, that's
So
right.
who's going to fill the need? So you get into these critical paths. And to be frank, I've never understood why they haven't gone down, in which it appears they are, to this model now in the screening assays.
Yeah, that's right. That's right. Well, you know, there are all kinds of reasons why, you know, you would adopt a new approach or not. But me personally, I think with the FDA's announcement earlier this year was the last hurdle. You know, for example,
Right.
if I'm developing a model to look for a new drug, say for Alzheimer's, um, you know, there is no good animal model for Alzheimer's. So with the FDA now saying, "Hey, we'll look at any model that you're showing us is predictive and has been validated, then we'll be happy." That just kind of takes that shackle off and lets me then pursue the way I want to pursue.
Right.
So
Right.
I think this year is really that. I don't know. I certainly did a kind of a nice relief breath and said, "Ah, now we can really start to move forward."
Yes. Any other questions before we end? Yeah, I think like I said, it's one of those crisis things, you know, there's opportunity, there's threats and there's opportunities at the same time, but
That's right. That's right.
So if there aren't any other questions, we can end. It's just about noon. Jay, thank you again. Very informative, and it's great to catch up to see, you know, since you started there, I haven't talked to you in a while, to see
Yeah.
where it's come, and it's really exciting. And the other thing, just to let people know, if anyone has an idea if there were where I should recruit somebody to give one of these webinars, or they have a personal interest in presenting a webinar, please just contact me directly. And again, thank you very much, Jay. And it's great to hear the exciting times in Manchester. I went to college in Manchester, New
There you go.
Hampshire. So,
Well, thank you, Bob. I really enjoyed it, and looking forward to talking with you further.
all right, good. Thank you all.
Bye.
Bye.